Hi, Siyu.
Hi, Aaron. Can you hear me?
Yep, good to see you.
Good to see you. So, shall we check whether the
Yeah, yes, please.
yeah. Let me see whether I can share the screen with you.
Hi, Pei-Lin.
Hi, Aaron.
Good to see you.
Yeah. So could you hear me okay?
Yes, perfect. Siyu is trying to share her slides to test them.
All right.
Do you know how to share them, Siyu?
Yeah. Does it work?
Yeah, it says it's loading.
Oh, okay.
I see a paper.
Let me see. Sure is paused. So, let me start share and then start play the PPT.
It's still showing this, your Acrobat reader. Can you go to your PowerPoint? Or just share your entire computer and then go to the PowerPoint.
Let's go share. Stop share. Why is it?
Or close everything else on your computer except your PowerPoint.
Okay.
I have that same type of headset for when I'm playing video games online.
This one make me feel safer.
Mm-hmm.
Let me see, share screen. And I just didn't come out. This one's no.
Yeah. Okay. So click that. Good.
Okay, I have two screens, so I think.
Oh no. Okay. So what you need to do is you need to click this. Stop sharing. Click that slideshow to play, and then start sharing. And then go. Do you know how to do that?
Let me take a look.
So you have to start the slideshow and then go back to Zoom and then do you know how to go back to Zoom and then, without quitting the slideshow and then do share? Or just share your whole computer?
So, no, I think it's still.
Okay, so
still
now
share the other screen. What?
No, but don't quit it. Just go back to that and we can switch it over. Perfect. Yeah, that looks good.
Oh, okay. Now it's work.
Can you just do a couple slides? Yeah, looks good.
Sure.
Okay, great.
Okay.
Okay. Pei-Lin, do you want to test your slides?
Yeah, sure. Could you see the slide okay?
Yeah, looks good.
All right. Let me check. All right. Okay, cool.
How's Taipei doing?
The weather is unpredictable, but today is fine.
Haven't seen you for a long time.
Yeah. So how's everything?
Yeah, we're good.
Okay.
Hope sometime we will meet in some place, in mainland or in some other places.
Would you guys attend?
I have like several years overlap with Pei-Lin when we were in UC Berkeley.
Oh, great, great.
Yeah, we definitely should
Hi,
catch up.
Zhang.
Okay, Zhang. So Zhang, after all that, our big meeting is going to be postponed.
Oh, sorry, yeah.
Yeah, I hate COVID.
Next month maybe just because it's so accidental, so everything pops out. It's weeks, right?
No. Yeah. The lesson is like never schedule a meeting when there's an important Chinese government meeting in Beijing happening in the same week.
That's part of the reason. That's part of it. So next month will be fine. I bet April it will be fine, definitely. Yeah.
Or maybe May. I'm still into it'll be good.
Yeah, we were on call anytime. Yes, no.
So I'm hosting a huge ALS meeting in Shanghai, and I had like a big fancy dinner scheduled. I had like the best food for our lunch. Anyway, but we'll have to postpone it. And the best speakers around China coming.
Yeah. It's just so accidental. Just a few weeks, right? So a few friends of mine came to Iowa and actually got meeting from Beijing. So yeah, I Xiaoqun were one of them. So when I come back, there's some come out. Oh,
yeah.
Yes.
Actually, the other lesson is I should just as soon as it's clear, just schedule a meeting within like a few days rather than I waited like 3 weeks rather than. Right.
1 week at most. So everything come out. Yeah. So Hangzhou will come here right away, right?
Yeah.
So it's very accidents come out.
Yeah.
But we'll cancel all the meeting in May already. So all the meeting
in May is canceled.
Yeah, so for me, so a lot of meeting in May. Oh, sorry, no, March. So our town is out.
Oh.
But for April is still there. So I'm going to Xiamen for the meeting, cell biology meeting. Yeah. So hopefully.
So maybe it's because it's not academic. Like my wife travels every week for work, and she gets COVID tests like someone giving her a COVID test every few days. So she's traveling all over China.
Yeah, as long as the private company is okay, it's where's it go? But university has a base of law.
Yeah. Oh, Pei-Lin. So I'm wearing my Neuro Zoom hoodie, and then in your honor, I'm wearing my Taiwan badminton Olympic.
All right.
I don't know how badminton works, but this is like a gold medal shot.
Yeah. Welcome to the team.
Aaron, it's prudent to stop there.
What's that? Oh, I'm not taking anything else off.
Oh, okay.
Taiwan court.
Good to know.
Sorry, Kong.
I thought about where the new Zoom to the IRS meeting, right? So
yeah, you should. So I've never played badminton, but I don't know what this means. But it says Taiwan court 1 in, and then I guess there's the ball.
Yeah, because of that, we won the Olympic and badminton games.
Yeah. I'm trying to play badminton around here, but I haven't played yet. How's Stanford come?
Stanford is fine. We are, so the Brown auditorium is being taken down. So there's a tremendous amount of noise from it's like literally 10 feet from my office. So
the college student back for college students.
No, it's just new construction. Oh, the most of the undergrads are back. So we're all having like in-person lectures now.
Right.
It's very it feels alive.
Great
again.
news. Yeah. Great.
Hi, Pei-Lin. Good to see you. Yeah, it's good.
Yeah.
I think the mask mandate is going away as we speak. It feels like it's back to normal.
I worry, like, I felt like that last summer, and then it all came back.
Yeah, I'm optimistic. I think it won't. Yeah, I think this is for real now.
All right. Good thing there's no other world problems other than COVID right now.
Oh yeah, right.
All right. Should we get started as well? Okay.
Yeah.
So, hi everyone. Thanks for coming for another week of NeuroZoom. Looking for International Women's Day. So happy International Women's Day. And next week we have two great speakers. Haining Zhang from the Volum Institute and Shuo Ling from National University of Singapore. But notice here, there's a new time starting next week in 8 a.m. in Asia, because in America, we switched to daylight saving time. We don't do that here in Asia. So Beijing, Shanghai, 8 a.m. Tokyo, Kyoto, Nara, 9 a.m. Taipei, 8 a.m. Singapore, 8 a.m. So a little bit earlier. But I think that's why Pei-Lin wanted to come today, so she didn't have to wake up early.
Yeah, I'm not a morning person.
Yeah.
Sorry.
Okay, so it's my great pleasure to introduce our first speaker, Dr. Pei-Lin Zhang from Academia Sinica in Taiwan. She did her undergraduate degree in Tainan, which is the south, so like Nanjing in China is the south, and Beijing is the north. And then for her PhD, she went to Taipei, like north, for National Yangming University. Here she did her thesis work on transcriptional regulation of tumor suppressors. She then came for a postdoctoral fellowship to UC Berkeley. It's one of the best public universities in California. And she did a postdoc with Mu-Ming Pu, who we all know. And here her research focused on neuronal polarity. How does the neuron establish axonal and dendritic domains? So like if you culture hippocampal neuron, say in culture, it starts out sort of unpolarized and there's little neurites. And it's a question of how one of those neurites decides to become an axon while the other ones become dendrites. And there's evidence that polarity of stabilization of microtubules plays a role in this. You can also do it by restricting polarizing factors to the future axon, or selectively stabilize other proteins. But in a very important paper in Neuron during her postdoctoral fellowship, Pei-Lin discovered another mechanism in which you could polarize a neuron, and that's by selective protein degradation. She discovered that the E3 ubiquitin ligase SMURF1 regulates axon formation by switching substrate preference from the axon determinant PAR6 to the growth inhibitory row A, small GTPase. So row A gets degraded, PAR6 becomes stabilized. This results in axon growth. And then she showed that this is induced by extrinsic cues. BDNF activates PKA, which phosphorylates SMURF1. She continues this work in her own lab at Academia Sinica, where she's an associate research fellow. She works on axon development, mechanisms underlying axon versus dendritic identity, and she's recently been exploring the role of selective transport of proteasomes to regulate this polarization. So looking forward to hearing her latest. Thanks, Pei-Lin.
Okay, thank you. So let me share the screen. All right. Could you guys see the slide okay? Right.
Yep.
So, okay. So thank you for the kind introduction and inviting me to share my current work on this fantastic platform. And most of the content in my talk today is unpublished data. So today my talk is all about the neuromorphogenesis. What drive and control the precise neuromorphogenesis always amazed me. So in the rodent cortical neuron or the hippocampal culture, the axon and dendrite polarize in their morphology, function, and the intracellular organelles trafficking within 1 day or two. after operating on the substance in vitro. And the precise control is quite robust. And the specialized membrane domain axonal segment were formed in day 5, and then proceed to another level of cell-cell interaction, forming myelin sheet when membrane of the oligodendrocyte contact or wrap the growing axon. And all of this robust multiple step of morphological transformation happen in an extremely soft mechanical environment. So our brain is the softest organ in the body. There's some open question I won't go through today, but we already have some answer for it. For example, the stiffness actually will not increase as the cell density increase, because cell itself is the softest element in the tissue, and even glia cell are softer than the neuron. And we already know that cells sense the mechanical gradient by track channel or piezo channel, and by changing the stiffness of the substrate, human mesenchymal stem cell could direct along neuron muscle or even the bone lineage. However, what is the underlying mechanism by which the brain softness are able to determine neural morphology is still unclear. Oops. So if we culture the neuron on a rather soft environment, it actually can grow fewer neurons, but stronger synaptic response. While the regular Banker's culture is roughly 1 day delayed in reaching the same extent of polarization. So, so if we, oops, yes, sorry, the slides is.
Sorry, I'll fix it. I'll fix it.
All right. Is it now okay? Okay. So yeah. This powerful effect in determining the number of neural protrusion, theoretically, the membrane tension played an important role. So the membrane tension can serve as a global inhibition for the neural protrusion, but at the same time, the local tension is increased as the neuron protrude. So in this sense, if we grow cell or neuron on a soft substrate with the low adherent strength and higher membrane tension, that can limit the number and the position of the neural initiation. In parallel, this protruding event can also confer a narrow tip structure where the short half-life GTPase or other neuromorphogenesis factor are able to accumulate or amplified. So this kind of structural activity feedback loop essentially similar to the diffusion reaction systems. So then if we relieve the membrane tension by treating cell with the myosin 2 inhibitor, then we will expect the overgrowth of the neuron, and which is true when we culture the neuron on the regular petri dish or cover slip. As you can see that after inhibition of the myosin 2, there's more minor neuron protruding out, will have the curved structure of the axon and craft of the growth cone. But it's not the case when we grow the neuron on a rather soft substrate. So usually the morphology of the growth cone remained intact or less altered or even not shortened after treatment by this myosin 2 inhibitor. So then we go for examining the molecular positioning of the cytoskeleton protein, such as the actin bundle and the tuburin bundle, and also the myosin 2 motor. And we found that a distinct distribution of the cytoskeleton components of the growth cone on the soft substance with the highly overlap actin-rich P domain with the tuberine terminal C domain. And if we compare with the regular culture on cover slip, we see a rather spreading out growth cone structure. So how this fits like architecture good at or can convert to a function. And essentially the growth cone is not only the moving front end of the neuron, it also serves as a force generating machine. So as you can see in this tension force microscope image, the tension force dynamic are able to monitor and detectable around the cell body, as well as the growth cone. So then we go on to compare the force generation under as different kinds of the condition and also the different stimuli. So the first surprise is that force generation activity of the growth cone is independent of the source signaling, which indicates that the force generation of the growth cone on the soft substance require only very minimized the focal adhesion, as the SRC inhibitor PP2 barely cancel out the force generation. And the second surprise is that chemotaxis or the action of axon guidance factor BDNF is able to generate force. As you can see that after best application of the BDNF significantly increased the overall force generation by the neuron culture on the soft substance, while the inhibition of the track receptor cancel the force. So when we see this phenomenon, we are thinking about what's the underlying molecular mechanism. So we resonate that the molecular mediator of the force and the signaling pathway used for chemotaxis were different from the neuron culture on a different substrate. The BDNF coated stripe significantly can attract the growth cone, no matter it's grows on the soft environment or the regular cover slip, with the most around 70% of growth cone returning toward the BDNF stripe. So when compare the protein distribution on the side of the growth cone facing to a stripe versus a west stripe, we can observe a preferential correlation between a growth cone transition zone factor dribbling with the adhesion molecule Pei-Lin facing the BDNF stripe on the soft substance, suggesting that asymmetric exposure to the BDNF on the brain soft environment are able to promote the formation between the molecule that are able to generate force. And we do confront this interaction with a biochemistry assay, but I won't show the result here. Instead, I show the result of this proximal ligation assay. Is an antibody-based in situ detection for a protein-protein interaction. So through which we can visualize the interaction between the T-zone factor dribbling with the adhesion factor Pei-Lin directly. And in this assay, the number of the PLA contact reflects the interaction of Pei-Lin and dribbling. So without the best application of the BDNF. And here you can see that the PLA single contact in the growth cone, and the contact density can further be enhanced when we treat it with the BDNF and eliminate when we inhibit the BDNF signaling. This is also true that the BDNF induced growth cone turning on the soft gel is again independent from the SARC signaling. And this finding indicates that the molecular mediator in the growth cone link chemical gradient signal to generation of the physical force, which then are able to drive the motion of axonal tip like soft environment. While the growth cone behave on the stiff substrate, but still mainly relies on the focal adhesion to exert actions. So to assess the function of the growth cone fourth generation molecule in terms of the axonal outgrowth of the neuron in the neonatal cortex in vivo, we use the in vitro electroporation to express the construct encoding small helping RNA against this dribbling protein in the subpopulation of neuron progenitor of the 17 rat embryo, and to enhance the analysis of the growth cone phenotype, we obtained the brain slice from the newborn P3 rat cortex when most the commercial axon already crossed the midline, and nearly 100% of the newborn cortical neuron at P3 already arrived at the cortical plate layer 2-3, exhibit the neuron bearing polarized phenotype with the dendritic arbor oriented toward the PL surface and the axon oriented radically in the cortical plate. And at the same time, most of the control commercial axon have reached to the contralateral site of the cross-chromosome, but this dribbling knockdown axon actually inhibits retardation of axon growth after crossing the midline, and with the quantification that indicates that it's about 20% on the contralateral side of chromosome, and consequently the extent of the branch of the innervation of the dendritic of the dribbling knockdown axon to the contralateral cortex layer 2-3 at the P16 was significantly less than the seen in the control axon. So this result then suggests the T-zone factor are important for the proper growth and the targeting to the axon projection of the neurodegenerate cortical neuron in vivo. So to summarize this part, the formation of the major force generating component and the functional structure of axonal growth cone can be properly formed only in the extremes of mechanical microenvironment. And the axonal path finding in its most energy efficient forms should be instructed by the chemical and physical contact of the tissue environment, contrary to this long-range neuron targeting to the designated brain area. So in the last 3 slides, I would like to share with you how robust this soft mechanical microenvironment can promote the oligodendrocyte precursor differentiation, as well as the myelination. Here you can see on the left-hand side is the human iPS-derived OPC culture on the tissue culture dish. And we can tell from the morphology that all of them maintain undifferentiated. But if we co-culture them with the neuron grows on different stiffness substrate without editing any glial differentiation growth factor or inhibitor, we are able to observe this spontaneous differentiation of the oligodendrocyte precursor into oligodendrocyte. And even on this brain soft substrate, the intact myelin sheets are perfectly formed. And all this entire process takes only 1 week to happen. So here is this transmission electron microscope. We have confirmed the formation of this competitive wrap myelin lamina in this prime form. And we are also able to detect the prior node and the node assembly by the marker Casper staining or the Ccntc staining that's indicating the fully mature stage of this myelinated axon on this prime 4. So we now use this prime 4 to screen out some interaction between the immune cell and the central nervous system. It's quite robust and powerful, able to dissect the cell-cell interaction and the individual cell behavior in vitro. And because in my lab, the other projects go for the proteasome function. And as we note that the proteasome serves the major machine for antigen presenting. So combined this in vitro prime 4 and with the another compresses activity, we are able to go for other aspect or project to study the axon biology or axon pathology. Okay. So with that is my talk today, and I would like to thank all my lab member and Lucy, Zheng, and Cheng contribute the project in my today's talk, and several collaborators in other institute, and also the funding resource. And I'm happy to take any question. Thank you.
Thanks so much, Pei-Lin. We have time for questions. Ka?
Pei-Lin, that's really interesting. So I guess these data would, makes me think that we should re-examine a lot of the mechanisms that, was based on the experiments done on high, cultural conditions that are really high, strong with a strong force, right? So on cover slips. So for example, have you looked at, the Frank Bradke's data on, depolymerizing actin or polymerizing microtubules can force that minor process to become the axon in the soft cultures?
Yeah, that's true. So I think the major problem to culture the neuron on the cover slip or the over-coded substance is that the oldest functional machine that should designate to either force or the other titled aspect being forced to participate in focal adhesion. So in that sense, if we relieve the strength of the focal adhesion, they will reconstitute this molecular mediator's function. So no matter from which point, if we reduce the membrane tension or adhesion, will promote the neuritic growth or axonal growth. So that truly will bias the conclusion of this molecule's function. And we frequently see this happen. In the lab, our model molecule is Pei-Lin. So the Pei-Lin involved in migration and adhesion to in other kinds of cell type, the fibroblasts. Mm-hmm. But in the neuron, it played a very minimized role in the focal adhesion. And actually, this adhesion molecule, for example, the vinculin and other other complexes involving the adhesion express very little in the neuron.
What really caught my eye was that the abundance of actin structures in hard cultures and the lack of those philopodia that is very much enriched of actin in your soft cultures. This reminded me of the results that Frank Bradtke many years ago, when he was working with Carlos, showed that if you depolarize the actin, then you can actually or depolarize the actin, make it more dynamic, then you can make the growth cone becomes the axon. I just wonder whether you have repeated that experiment in your cultures. Yeah.
I think if I do it in the soft gel content, I might not able to see this robust phenomenon because the actin already
is not very strong there.
Yeah.
Yeah, got it. Okay.
Yes.
Okay, great.
Yes.
Thank you.
Other questions? Okay. If no, actually, one question for me, for the myelination story, do you see in situations if you injure or demyelinate an axon, if you see similar dependence on the soft surfaces for remyelination?
We haven't done it yet because this in vitro plan for we still need to more carefully characterize it. But we already done some other experiment in terms of the T cell killing assay. So it indeed can induce the apoptotic and also the damage in terms of this myelin sheet and as well as the OPC cell. Yeah, so we are going to do that to with the more careful characteristic of this platform.
So maybe if there's no more question, I can ask one more. So if integrin is not that involved in the force sensing, then what is sensing the force?
I think the membrane channel itself are able to do that.
Mm-hmm.
So when they
which channel?
Yes, a good question. We kind of in the cortical neuron, we kind of rule out piezo1 and 2, but the trap channel might play a role. But
those trip channels,
yeah,
those initial papers, there are hardcore studies on these trip channels, and the evidence that they are mechanical gated is very weak, in my opinion.
Yeah. Yes. So it's kind of I'm also thinking of that under very extremely soft environment. Might be the internal dynamic of this fourth generation is already sufficient to drive the growth of the axon. It's not necessarily need strong adhesion, so it can preserve all this sensing machine for the chemical guidance. So the soft environment essentially just provide a permissive environment to increase the sensitivity of the chemical guidance.
Okay, thanks so much, Pei-Lin. Zhang, you're up.
Sure, great. Thanks, Pei-Lin. So it's my great pleasure to introduce our colleague Siyu Zhang, who is now assistant, I believe soon will be a associate professor in Shanghai Jiao University School of Medicine. We're nearby. So Siyu is one of the awesome ION alumni. So I remind me how long I have been here. So when I just come to ION, Siyu and her husband Ming as the students. So Siyu has a bachelor degree in Nanjing University, and then she went come to ION to work with Mo Mingpu and Xiaohui Zhang. To Cheng has become a super expert in electrophysiology. And so after PhD in ION, she went to UC Berkeley to work with Yang Dan, where she's working with the visual system to studying the neurocircuitry, where he has a amazing story of finding out the long-term, long-range cortical top-down regulation involving attention. So in 2016, she come back to China along with her husband, started her own lab here. So Siyu had continued her study in the visual system, so where he has a amazing story coming up. So studying the also the hierarchy processing in the cortical mediated by cortical interneurons. I believe that this is where Siyu will talk about today. So okay, so with that, so welcome, Siyu, take it away.
Thank you. Thanks for the nice introduction. Let me share my screen. Okay, is my screen okay?
Yeah, please go ahead.
Okay. So thanks for the invitation. I'm really excited to share my story in your Zoom. My work is about how the hierarchy in sensory processing reflected by innervation balance on cortical interneurons. Hierarchy is a central feature in the organization of neural networks. Functionally, the sensory systems are hierarchically organized. In a visual system, visual stimuli generate bottom-up information flow from the retina to LGN to V1 and then to a series of higher-level areas. Neurons in the high-level areas have larger receptive fields and encode more complex features. So this is one direction, bottom-up, and there is another direction, top-down. The high-level areas make behavior-related top-down modulation of visual process. So the hierarchy of each node in this network determines the direction of the information flow between them. Previous studies in primates have shown distinct axon distribution patterns of long-range cortical inputs with different functions. Axons from bottom-up inputs mainly located in layer 4, and axons of top-down inputs are in superficial and deep layers. In 1991, Pei-Lin and Van Essen generated the hierarchical organization of primate visual system based on the axon distribution patterns. Around the same time, Burkharter's lab found conserved axon termination patterns of CC inputs in rats and defined the areal hierarchy of rat visual cortices. Recently, a series of significant progresses have been made in the hierarchical organization of the mouse brain. In 2017, Austin's lab proposed a hierarchical organization of mouse brain based on the distribution of inhibitory neurons. They quantified the distribution of major inhibitory neuron types, including PV, SST, and VIP neurons. These interneurons formed conserved circuit motifs. In this motif, PV and SST neurons inhibit pyramidal neurons, and VIP neurons inhibit SST neurons to disinhibit pyramidal neurons. Based on their quantification, they found a cortical organization principle. The sensory motor areas with low level of hierarchy are dominated by PV neurons, and the frontal areas with high level of hierarchy are dominated by SST neurons. They then rank the hierarchy of mouse cortices based on the ratio of relative PV and SST density in each area. And Allen Institute has done a series of nice work on this topic. Based on the axon termination patterns of cortical CC and cortical TC target lamina patterns, they expanded the hierarchical organization to all the cortical and thalamic areas. So first, they collected the axon termination patterns of more than 1,000 tracer experiments. These are the patterns of CC inputs and TC inputs. And then they quantified the projection strengths by cortical layers and perform unsupervised hierarchical clustering and find the 9 common patterns. Finally, they build a model to assign connection patterns as either feed forward, bottom up, or feedback, top down. So after identification of the direction of each projection, they generated testable predictions of hierarchical positions for individual cortical and thalamic areas based on the directions of all the inputs and outputs from each area. So here are the hierarchy of the visual cortical areas, and this is a hierarchy of the cortical modules revealed by their algorithms. And they immediately tested the cellular level functional interaction in the mouse visual system across the anatomical hierarchy. Using NeuroPixels probes, they simultaneously recorded neuronal spikes in 6 cortical and 2 thalamic visual areas in the mouse brain responding to visual stimuli. They found that the visual stimulation generated strong bottom-up wave of activity propagate up the anatomical hierarchy. Wave-1 neurons lead the activity and AM follows other regions, indicates that wave 1 is at the lowest level and AM is at the highest level of hierarchy. In addition, all 4 popular measures of hierarchical processing changes across the anatomical hierarchy. The neurons in the areas with higher level of hierarchy have longer response latency, larger receptive field size, and less phase locking to drifting gratings, and longer response decay time. So their results demonstrate that the functional hierarchy mirrors anatomical hierarchy. So now we know the hierarchy of the mouse brain and the direction of the long-range inputs. To serve their functions, the directional long-range inputs need to interact with the different types of neurons in their target areas. But it is still largely unknown how this interaction across different hierarchical stages. To answer this question, we systematically analyzed the anatomical and physiological properties of long-range inputs to the 3 major types of interneurons at different hierarchical stages of visual processing. So first, we characterized the whole brain inputs to cortical interneurons. Second, we identified the anatomical rules. And finally, we validated the anatomical rules with functional connectivity. Let me start from the first part. In this part, we use rabies virus assisted mapping. Using rabies virus assisted mapping, we systematically characterized the whole brain inputs to the 3 major types of cortical interneurons, PV, SST, and VIP neurons at different hierarchical stages of visual processing, including the primary visual cortex, VIS, the medial association cortices, RSP, PTLP, and the frontal cortices, ACA, POLA, and ORB. We use a customer written software package to analyze the digitized brain image. This software is based on the Allen mouse brain atlas. After reconstructing more than 100 mouse brains, we generated this quantitative brain-wide input matrix for cortical interneurons. Each row contains the input percentage averaged from experiments in one target region, and columns show the input errors. Our relabeled presynaptic neurons were found in multiple cortical and subcortical regions. The RV tracing has shown that the local interneurons shared all the major long-range inputs with biased input percentage, and most extensive inputs were from other cortical areas and thalamic nuclei. We identified the major CC and TC inputs to cortical interneurons. Left panel shows the major CC inputs with more than 2% input percentage for at least 1 interneuron type. And the right panel shows the major TC inputs with more than 0.5% input percentage. After characterization of the whole brain inputs to cortical interneurons, we asked whether there is an anatomical rule to link the hierarchy to the input patterns of CC and TC inputs on cortical interneurons. So in this part, we mainly use the support vector machine SVM classifier to review the rules. Since the cortical areas examined in the current study belong to the visual, medial, and the prefrontal modules with well-accepted intermodule hierarchy, the CC inputs across these modules were considered as well-defined bottom-up and top-down inputs. So notably, the hierarchy of the cortical areas in the same module varied in different studies due to the different criteria used. So we put the intramodule CC input aside for now and check them out later. To understand the relationship between the input directionality and the input patterns on interneurons quantitatively, we use the data from well-defined bottom-up and top-down CC inputs to train an SVM classifier. Here, panel C shows the cross-validated classifier performance. Gray area indicates the 95% confidence interval of classifier performance on shuffle data. So we can see that only the classifier trained with a combination of input percentages of PV and VIP neurons shows the performance above the 95% confidence interval, indicating that only this classifier accurately predicts the directionality of well-defined bottom-up and top-down inputs. And panel B shows the decision boundary, the black line, for the classification according to the PV and the VIP input percentage. Red stars are the well-defined top-down CC inputs. Blue stars are the well-defined bottom-up inputs. They were nicely separated. And this classifier also predicts the directionality of the intramodule CC inputs, the red and the blue circles. These results indicate that the link between the hierarchy and input patterns is specifically reflected in the relative weights of innervation on PV and VIP neurons. We thus defined the hierarchy index HI anatomy for each long-range inputs as the input percentage on VIP neurons minus the input percentage on PV neurons and divided by the sum of them. Well-defined bottom-up inputs show negative HI anatomy values, which means these inputs prefer PV neurons. In contrast, well-defined top-down inputs show positive HI values, indicating their preference on VIP neurons. The SVM classifier trained with HI anatomy values was called classifier anatomy. It revealed that the decision boundary for classification of bottom-up and top-down inputs is close to 0, the dashed line here, suggesting that the sign of HI anatomy can be used to predict the directionality of a specific input. And notably, the reciprocal intramodule inputs, the circles with the same colors here, they show opposite preferences for PV and the VIP neurons, indicating opposite hierarchical directions of these inputs. We then computed the hierarchical score of each cortical area based on the CC connections. We adapted the algorithms in Allen Institute's 2019 Nature paper. In short, the initial hierarchy score for each cortical area was defined as the average differences between the HI values of its outputs and inputs. The score of each area was then iterated on the basis of the current hierarchy scores of its targets and sources to further refine the hierarchy. In this way, we obtained the hierarchical organization of cortical areas in the visual network. POLA is at the top and VIS is at the bottom. And here arrowheads denote the direction of each inputs. Red lines are top-down inputs and the blue lines are bottom-up inputs. Now we take a look at the TC inputs. Allen Institute's work revealed that the axon termination patterns of hierarchically organized TC inputs obey similar rules as CC inputs. Thus, we hypothesized that the anatomical rules derived from CC input patterns could be used in TC input to predict their directionality. To simplify our model, we grouped the thalamic nuclei, then provide major inputs in the ventral, lateral, anterior, medial, midline, and intralaminar groups according to Allen-Brown atlas. We calculate the HI anatomy of TC inputs in the same way as CC inputs and use the SVM classifier trained with CC inputs, the classifier anatomy, to predict the directionality of TC inputs. The top-down TC inputs are in the pink area and the bottom-up inputs are in the blue area. To test our hypothesis, we added the directionality data of TC inputs into the cortical network and examined whether the TC inputs affect the cortical hierarchy inferred by our algorithms. We compared the hierarchy constructed from both CC and TC inputs with that obtained using only CC inputs. Pink is the CC only network, and the purple is the CC and TC network. These two versions of hierarchy has similar global hierarchy scores, and adding TC connections did not change the ranking of cortical errors in the vision network. The hierarchical ranking of cortical errors showing in pink is the same as showing in purple. So this indicates that the CC inputs provide a similar hierarchical information to TC inputs. This is a schematic diagram summarizing the hierarchical organization of the vision network. The yellow dots are cortical errors and the blue dots are thalamic errors. The arrowheads denote the direction of each input. We found that the hierarchy reflected by the input patterns of cortical interneurons is naturally consistent with the one derived from axon termination patterns. In both versions of the hierarchy, thalamic errors are found at the top and the bottom, and the cortical and other thalamic errors are in the middle. And finally, we validate the anatomic rules with functional connectivity. We use optogenetics-assisted electrophysiological recording to investigate the synaptic strengths and dynamics of major CC and TC inputs revealed by RV tracing. These synaptic properties are crucial for understanding the function of a specific input. So we focused on comparing the synaptic properties of different types of interneurons in layer 2-3 because VIP neurons are mainly distributed in layer 2-3 across different cortical areas. So first, we measured the strengths and dynamics of well-defined bottom-up and top-down CC inputs. Here are the virus injection sites and the axon distribution patterns in the target areas. So these are two examples. For the bottom-up inputs from VIS to SAA, we can see that the input strength is strongest on PV neurons and the pair-pulse ratio is highest in VIP neurons. And in contrast, for the top-down inputs from SAA to VIS, the input strength is strongest on VIP neurons and the pair-pulse ratio is highest in SST neurons. This is a summary for all the intermodule inputs. The bottom-up inputs to PV neurons exhibited the largest amplitude, but rapid depressing of synaptic potentials, whereas the synapses onto VIP neurons are initially weak, but showing synaptic facilitation. In contrast, the top-down inputs onto VIP neurons were strongest but show depression, and those onto SST neurons were initially weak but show facilitation. Thus, well-defined bottom-up and top-down CC inputs preferentially recruit distinct types of local interneurons. Next, we measure synaptic properties of intramodule CC inputs, and we found that in each pair of reciprocal connections, one is strongest on PV neurons, similar to well-defined bottom-up inputs. The other is strongest on VIP neurons, similar to well-defined top-down inputs. Last, we calculated the HIEs based on the synaptic strengths. Use the same equation for the anatomic input percentages, and then we train the classifier ifs using the HIEs values of well-defined bottom-up and top-down CC inputs. The dashed line here shows the decision boundary revealed by classifier ifs. It separates the long-range CC inputs into two distinct groups identical to them determined by classifier anatomy. Moreover, we calculated the hierarchy score of cortical errors based on HIEs. This version of hierarchy provided an increased global hierarchy score and a similar ranking of cortical errors. The green dots has a similar sequence as pink dots. So together, our results also show that functional hierarchy mirrors anatomical hierarchy, but from a different point of view as Allen Institute's work. We next measure the synaptic properties of TC inputs. We focus on the inputs from association, nuclei, MD, and LPLD, which are known to be involved in visual processing. MD only innervates frontal cortices. The synaptic properties of MD inputs to the frontal cortices are similar to the bottom-up CC inputs, strongest on PV neurons and facilitating on VIP neurons. LPLD are the only thalamic nuclei innervating cortical areas at all stages in the visual network. The synaptic properties of LPLD input change markedly according to the target. Similar to bottom-up inputs, LPLD to ACA inputs was strongest on PV neurons. On the contrary, LPLD to VIS inputs was strongest on to VIP neurons, consistent with top-down inputs. And notably, LPLD to RSP input was strong on both PV and VIP neurons, exhibiting the characteristics of both bottom-up and top-down inputs. Then we apply the classifier ifs trained with CC inputs to the TC input tested above. The classifier ifs well separated the bottom-up and top-down TC inputs. LPLD to RSP input is strong on both PV and VIP neurons and is thus close to the decision boundary. Together, our results on both CC and TC inputs show that the preference innervation pattern on TV and VIP neurons is a critical feature in the hierarchically organized thalamocortical network. Last but not least, we examined the synaptic dynamics linked to the directionality of CC and TC inputs. We can see that the bottom CC inputs are facilitating on VIP neurons and depressing on other types of neurons. In contrast, the top-down CC inputs are facilitating on SST neurons. However, the TC inputs are always depressing on SST neurons. Statistical analysis has shown that the bottom-up CC inputs, the blue bars, have significantly lower purpose ratio PPR on SST neurons and a higher PPR on VIP neurons than top-down CC inputs, the pink bars. Furthermore, we use the synaptic dynamics information of both CC and TC inputs to train the SM classifier and found that with a single cell type, only the classifier trained with the VIP neuron synaptic dynamics significantly correlated to the directionality. The decision boundary revealed by this classifier is close to 1, the dashed line here, indicating that for both CC and TC inputs, the bottom-up inputs are facilitating and top-down inputs are depressing on VIP neurons. And this is the take-home message. Based on the anatomic and physiological data, we obtained a unifying rule for the hierarchical organization of cortical and thalamic connections from their innervation patterns to cortical interneurons. Bottom-up inputs favor PV neurons and top-down inputs prefer VIP neurons. And our model based on this rule generated testable predictions of hierarchical positions for cortical and thalamic areas and provide a basis for understanding the distinctive role of bottom-up and top-down inputs in the brain. At last, I'd like to thank all the lab members. Yan Mei and Guofeng are the first authors of this current work, and my collaborators, Dr. Ming Xu and Tong Li, ION, Fuqiang Xu in SIIT, and Tianle Xu in Shanghai Jiao Tong University. And thank you all for your attention. Now I'm ready for the questions.
Great, thanks you for so rigorous work in the lab. So some questions. I might start first one. So I'm pretty interested in the interneuron in the RSP, retrosplenial cortex. So for that, have you injected the critical virus or RB virus into the retrosplenial in order to find out the output, the upstream downstream connection yet?
Oh yes, we have injected the virus there.
Okay. So you haven't.
We have.
Okay. So that's why I find out where strong connection between the retrosplenial with the visual cortex. Right?
Oh yes.
But in particular, retrosplenial, could you evaluate which type of the interneuron may play a most important role regarding the SST or PV, or?
So according to our results, there's different types of interneurons play different roles in this hierarchical network. So PV neurons were recruited by the bottom-up inputs, and YP neurons recruited by the top-down inputs. And bottom-up inputs could also cumulatively activate YP neurons, and top-down inputs will cumulatively activate SST neurons. So I think it will be a dynamic network when the visual processing is ongoing.
Oh, okay. I'm gonna talk more about it. The reason I'm asking because in our particular, one of the high functioning autism mouse model, in SEP1 mutation, it shows up the increase in PV mutation, particularly in retrosplenial, but not SST. So your finding is suggesting there may be some bottom-up problem instead of a top-down problem, right? I can talk more about that.
Yeah, sure.
Great. Okay, more questions? Kevin, please. Kevin, you can ask yourself.
Sure. Yeah, so nice, very nice work. So I'm always interested in how the different type of interneurons, how they process the information and basically guides the local neural networks to proceed to process the information. So do you think this rule is also universal for the other brain regions, especially the other cortical regions, and also how this different type of a top-down versus a bottom-up mainly driven inputs will be modulated during different contexts, such as diseases and physiological conditions?
That's a great question. Thank you. So, so I think this cortical motif formed by these three types of interneurons is conserved across the cortical areas, which is also revealed by the data from Austin's lab. But there are some differences along these hierarchical gradients. For example, the SST neuron, the density of SST neurons is highest in the frontal cortical modules. And we also found that both bottom-up and top-down connections to SST neurons is stronger in these prefrontal modules comparing with in the other modules or cross different modules. And so in the disease or the other physiological conditions, for disease, I'm not quite sure, but for the other physiological conditions, the Komiyama's work have shown that learning is gonna bias this balance between bottom-up and top-down inputs towards the top-down. So, but I think it is still widely open that in different diseases or other physiological conditions, how's this balance between bottom-up and top-down modulation gonna change along the hierarchical organization, the line of hierarchical organization in the brain.
Thank you. Thank you very much. Very nice work.
Okay. Please, Dr. Xu. Yeah, I'm trying to spend.
Yes.
Yeah, go ahead.
Yes. So this is could you hear me?
Oh, yeah. Sure.
Yeah, so this is a very, very impressive work. So I wonder, would you able to provide some speculation or idea of why would this hierarchical, so functional implication? So basically, why would the brain organize this way? We'll have a differential thalamocortical and cortical input to different interneurons according to their hierarchy in the visual areas.
So according to our data, we actually thought that since the bottom-up and the top-down inputs are has a temporal delay, right? The bottom-up come first and the top-down come back. So according to the temporal dynamics of the bottom-up and top-down inputs on different types of interneurons, We think the bottom-up input's gonna first recruit PV neurons, and then later on it's gonna recruit VIP neurons. And temporally, it is probably synergistic with the top-down inputs onto VIP neurons, which will work better to activate these VIP neurons and facilitating the top-down modulation. And then later on, this top-down inputs gonna recruit SST neurons, which will inhibit both pyramidal neurons and other types of interneurons, which could generate a balance in the local network, which means rebalance the local network to make this network prepared for the next visual stimuli. That's our interpretation, but still need to be tested in vivo experiments.
Okay, great. More questions? Okay, no more questions. Thanks, Pei-Lin, for Siyu for very precise time today. Very long, great. Yeah. Okay. Thanks, Pei-Lin, and see you for great talks.
Thank
See
you.
you all next week for 8 a.m. So get up earlier. I have to start a new Zoom at home.
Thanks.
Right.
Thanks everyone. Yep. 8 a.m. next week in
See you
Asia.
next week.
All right. Bye everyone. Thanks to you. Thanks, Pei-Lin.